I'd like to welcome Dr. Pinto to the stage. He'll be presenting his video via pre-recording.
I want to thank the Focal Therapy Society Organizing Committee for this opportunity to speak on non-inferiority trials as it relates to whole gland therapy and prostate cancer. Sorry I couldn't be here at the FDA session, but we'll be here for the rest of the meeting and looking forward to seeing all my colleagues and friends throughout this meeting. As it relates to this lecture, I have no disclosures. Special thanks goes to Dr. Charles Heswani in helping prepare for this lecture. Charles is one of our SUO fellows, finished McGill University residency, really is doing an outstanding job with us, looking for an academic position starting in July in uro-oncology and also focusing on image-guided interventions and focal therapy. Just had a baby, starting a wonderful family, and he needs an academic home. So anybody looking to get some help in their department, please reach out to me. He is wonderful. So we'll focus on definition of rationale, reporting of non-inferiority trials, determining what's called non-inferiority margin, choosing endpoints, and then determining the inclusion criteria. Now, what's the rationale? So, if you have a new treatment that is felt to be sufficiently favorable by patients and physicians alike, that there's a willingness to sacrifice some degree of maybe efficacy, small amount, to receive the benefits of this new treatment, whether it's more improvements in morbidity, reduction in cost, more available to patients, better safety profile. These are factors that one would have to consider for a non-inferiority trial. Here are some of the pros and cons of such trials. First of all, it's okay if the comparator or the active control group is clearly more efficacious. Sometimes non-inferior trials actually show to have the new treatment being a superior cancer therapy. Of course, we mentioned could be better tolerated, more convenient, more available, and cheaper. And there were a few cons too. Sometimes the new therapy is ineffective at its treatment based on the criteria proposed in the trial. Failings can occur depending on trial design. And sometimes, although similar in their treatments, they're really not close to being comparable or identical for the treatment opportunities. Now, as you embark on writing trials, non-inferiority trials for focal therapy, it's important to be familiar with the Consolidated Standards of Reporting Trials, or CONSORT statement from 2010, specifically focus on the 2012 publication that focused on non-inferiority and equivalence randomized trials. In this regard, you will have a 25-item checklist for the objectives, outcomes, interpretation, reporting opportunities, and really a great place to start as you embark on this work. Now, there are potentially different outcomes in such trials. First one being a non-inferiority and superiority. That's where the 95% confidence interval all lies on the side for the new treatment and does not cross the odds ratio of 1. For a other outcome, which is a non-inferiority outcome, you see here that it does cross the odds ratio of 1 and it's within the margin, and we'll discuss the margin in the next few slides. Another outcome could be that the result is a non-inferiority and inferiority. This is unusual when this happens. It doesn't happen too often. Confidence interval is above 1, but still within the margin. For something to be a new treatment and be both non-inferior and inferiority, this is usually a study design issue. The margin that was set was too lax. It can be inconclusive, where it's crossing 1 and goes beyond the margin. And of course, then there's the potential that the new treatment outcome new treatment is just purely inferior, as shown here, and favors the traditional method that you're comparing it to. So let's spend a few moments on the margin, as this is very important to non-inferiority trials. The non-inferiority margin should be considered based on clinically acceptable information. This may come from experts, guidelines, prior trials. May not always be as robust as we like. Do the best you can. In particular, it should be a margin set close to standard treatment without loss of clinical benefits. Particularly, the FDA often works with the sponsor to set this margin. And the size of the margin is important. As you mentioned before, if it's too large, you'll end up with an inferiority. The outcome, if too large, makes it almost too easy. And what we call a permissive margin will guarantee a non-inferiority. Think of it almost like you have a parking spot for a large bus, a tour bus, and you want to put your little sports car in that. There's just so much space, it's going to happen for sure. So you have to set that margin a bit more tight. Really should be the largest difference between the new treatment, focal therapy, and the old treatment. In this case, that's still considered to be acceptable. And we mentioned the consort statement, very particular how we define and state the importance of different aspects of the trial. And you can see from this here, even when the margin justification is important and expected, often is not even given when these trials are created. And since this margin is so important to non-inferiority trials, two large regulatory agencies here in the States, the FDA, and overseas, the European Medicines Agency, have wonderful guidelines for clinical trialists to understand how to set that margin. I believe the European Medicine Agency is having a revised document relatively soon. The one from the FDA was done in 2016. And as you can see in bold font, we have to make sure that the margin does not exceed a clinically meaningful benefit that's been established by whatever information you have, historical data, expertise in the field. This is to maintain the new modality or new treatment's integrity and ensure that it's still effective. And let's say that historical data shows that standard of care treatment has a 20% reduction in a negative outcome compared to a placebo. A non-inferiority margin might be set to say 10%. It means to preserve at least half of this benefit from the standard of care. And specifically, say for prostate cancer, if radical prostatectomy surgery has a 90% survival rate, and you aim to preserve at least 80% of that benefit, then that margin is set at 10%. And here's an example from, say, the PIVOT trial. Trial that looked at surgery versus observation in the VA setting. If disease progression was the objective, in this case, almost 30% risk difference between surgery and observation, favoring surgery. If a non-inferiority trial would be used to compare, say, focal therapy to surgery, with the outcome also being disease progression, a margin could be set at, say, half that, or about 15%. Now, staying on this topic of the margin, how to set your margin, this also depends on the endpoint and its severity. So, for example, death from cancer, a very severe endpoint, then you want that margin to be tighter, closer to the odds ratio of 1. Let's say that the endpoint for the non-inferiority trial was the need for additional therapy or salvage therapy. Then maybe not as severe, the margin could be a bit larger. Also important to remember for the margin that it's a one-sided difference or significance. What does that mean? So a non-inferiority trial is saying that this new therapy is not worse than the standard of care therapy. Focal therapy is not worse than whole gland radiation or surgery. Therefore, really the interest in the difference between the new therapy and the established therapy is just a single one-way direction. Is it worse by a certain amount? And therefore only the upper bound of the confidence interval matters in this case, so alpha is cut in half. So here's an example to help understand this further. This is the Carmina trial, looking at sunitinib alone or sunitinib with surgery for nephrectomy, in this case for metastatic RCC. This is probably one of the most important non-inferiority trials in uro-oncology. So in this case, the primary outcome was overall survival. Again, this would be a very severe endpoint. So a tight boundary or margin. In this case, the non-inferiority boundary upper limit of a 95% confidence interval was a hazard ratio of 1.2. That's the margin it was set. For sunitinib treatment alone was 0.9, and therefore the margin was set 1.1, so well below 1.2. And as we all know from the study, sunitinib alone was shown to be non-inferior when compared to upfront nephrectomy followed by sunitinib. So now we'll move from the margin to the clinical endpoint for such trials. You need to have a sweet spot between these three: feasibility, clinical relevance, and its acceptance by regulatory bodies, whether it's US or overseas. This all being done to maintain the ethical standards we discussed before for such trials. Now focusing on clinical endpoints for focal therapy non-inferiority trials. These endpoints will depend on the comparator arm, the whole gland therapy arm you're comparing it to. And because it's prostate cancer focal therapy, short-term outcomes are likely more meaningful and obtainable. Considering the still relative experimental nature of focal therapy and the different modalities that are available, looking at outcomes that could take decades to mature, especially in prostate cancer, are likely to be a challenge and maybe not beneficial in this case. In this situation, then primary outcomes such as disease-free survival or time to salvage whatever therapy was performed relative to whole gland or systemic therapy would be, I guess, favored or privileged over overall survival or met-free survival. Now, staying on this clinical endpoints, it's important to remember that the endpoints chosen really depend on the comparator or the modality you're putting focal therapy up against. So, for example, if your clinical endpoint is persistent cancer, an in-field recurrence after focal therapy, that can really not be put up against whole gland surgery, for example. Now, if your endpoints are cancer-specific survival, metastasis-free survival, overall survival, then yes, you can compare focal therapy, if that's your endpoint, to any type of whole gland therapy. If you look at salvage therapy or retreatment rates for prostate cancer, and again, all these modalities are fair. Local control rate, disease progression within the area of treatment, again, you can compare it to many different modalities. If you're looking for that negative biopsy rate within the prostate itself, then you're really limited to comparing it to, say, external beam radiation or some type of other radiation, brachytherapy, proton beam, or something like that. Now, once again, the primary objective or the primary outcome is what will make or break your non-inferiority trial. But secondary objectives are important, often used in other trials, and in this case, no different. Reporting adverse events, functional outcomes, health economics such as qualities, rat path correlation study, tumor microenvironment, all can add to the trial. But again, for non-inferiority, the primary outcome or primary objective is going to be the most important. Moving to data analysis, there's different ways to do this. Intention to treat looks at those patients in the study group that they were assigned to, and really regardless of the adherence to the protocol, whether they complied with what was required of them or did they even get treatment. And often this is lauded for representing real-world settings. And in a per-protocol analysis, it's a bit more stringent. It looks only at those patients who completely followed the protocol of the study, really looking at ideal conditions, maybe less realistic. For these non-inferiority trials, analyzing both ways probably is the best. It gives you very strict criteria and analysis per protocol, but also it can look at it through the lens of a real-world experience. And remember, primary objective is the goal here. That's what you're going to publish on. Think of the Carmina trial. Secondary objectives are important, but do not influence the non-inferiority conclusion of the primary objective. Regarding study design, the new treatment, in this case focal therapy, the control arm, whole gland therapy followed by parallel group design. Now, you can't really blind the clinicians providing the therapy in this case. It's very different surgery, focal therapy, but if you can, others involved in the study should be blinded, like the radiologists, pathologists, statisticians, best you can. For sample size, it's not just calculating the difference in primary endpoints, but also that non-inferiority margin. That's why the margin was so important in this presentation, to ensure sufficient power to detect non-inferiority. And lastly, for study design, think of interim analysis, not just safety, but also short-term results. Have predefined stopping endpoints for interim analysis, for the safety of your patients. As we conclude with inclusion criteria, just a brief paragraph from the FDA. Remember, the choice of not using a superiority trial, but going with non-inferiority is actually an ethical question, because doing nothing or a placebo or very low dose of a drug, would be an ethical concern for those patients in such trials. So with that in mind, we have to be thoughtful as we design these non-inferiority trials for focal therapy. Remember, the goal is not to replace whole gland therapy, radiation, surgery, but to give urologists another opportunity to treat in a different way. As such, we're using non-inferiority trials to get to that goal. And we're all here. We've created the Focal Therapy Society in support of this work and making the lives of our patients better. But remember, the modalities are different. We have to, start slowly. So, arguably we could look at a lower risk population of patients where we're using our best modality, NPMRI, using targeted biopsies and systematic biopsies to fully risk stratify our patients. So we don't enroll patients on trials for focal therapy that are not good candidates for focal therapy because of cancers that are missed that are more aggressive. This intermediate risk population of patients is probably ideal to start with, making sure that it's not multifocal or at least not on both lobes. And because we have so many great modalities that can really treat prostate cancer in a focal way, go with what you're used to, what you have experience with, what you do well as you design these trials. So in summary, know your goals. Perhaps a bit too soon to go for superiority, equivalence of focal therapy, thus this talk focusing on non-inferiority compared to standard of care. Risk stratify well. In this case, it's kind of intermediate risk group, seems to be the ideal sweet spot for non-inferiority trials. Go with short-term survival endpoints as much as possible. Just the nature of prostate cancer makes it challenging otherwise. And this could pave the way for future long-term studies and maybe even someday potential superiority trials. Include a post-hoc analysis in the non-inferiority protocol because you never know. As we showed earlier in the talk, one of the potential outcomes is you could prove it to be superior. And it's just overall, we're not here to tear down and replace traditional whole gland prostate cancer therapy. We're here to provide other treatment opportunities for us in our patients' care. And therefore, these non-inferiority trials may provide that opportunity. Thank you for your attention, and really special thanks to my federal government colleagues from the FDA, who just like last year came in support of our Focal Therapy Society and share our mission of improving the lives of our patients with prostate cancer and finding new modalities, in this case focal therapy, in support of their treatment. Thank you.
So that concludes the didactic portion. The Q&A is going to start. I'm going to invite Dr. Susman and Dr. Anthonino up to the stage to answer questions.
And Dr. Klotz, too.
And Dr. Klotz. He's just going to the FDA. Fernando Bianco. I'm going to step down so there's more room.
Okay. So I could maybe start with a question for Dr. Susman, since you're first up. So one of the challenges for a lot of us in the focal therapy world is that while the Weinstock paradigm of using active surveillance as a comparator is described as the optimal design for trial, there's a lot of interest in treating what we would call more clinically significant disease. And so I wonder if newer tools like PSMA PET, some of the genomic tests that are available, which allow us in clinical practice to better risk stratify, better stage our patients, might open the door to inclusion of, say, GG3 disease or higher risk patients when they're properly selected in trial design.
Yeah, I mean, I think there's sort of two patient populations. One is the patient population that's appropriate for active surveillance, and the one is the patient population that needs treatment with whole gland therapy. And I think we're agnostic about how you define those populations, as long as it's well supported by those patients that are eligible or appropriate for that modality as the control arm. And use of newer technologies, PSMA PET, genomics, et cetera, I think would be part of that.
I just wanted to follow Dr. Samir's thought. The same question with respect to what the trial design that was posted, Dr. During your talk, when looking at that paradigm with active surveillance, is that where the FDA is starting from right now when you meet with industry, when they're looking for an indication for prostate cancer? Is that the foundation, or how are we counseling industry and investigators on developing trials that can get to device approval for an indication for prostate or label for prostate cancer?
So I'll just say, again, I'm speaking from the biologic and drug perspective. CDRH may have a different perspective, and certainly operate under different regulations for devices. The trial schema that I presented is just one potential approach that could be used. Again, we gave some of the logic around why that might work. In terms of the for an active surveillance population, if you're looking at a focal therapy, again with if you're comparing against whole gland therapy, again that may require a different trial design, and certainly there I put up some of the considerations and pitfalls that for that population.
Thanks.
I mean, certainly industry investigators are welcome to come up with an alternative, and we're very open to hearing. If you want to schedule a meeting with us, if you have a drug or biologic you're developing, please schedule a meeting. We'd be happy to talk about your an alternative paradigm.
You want to talk?
To follow up on what Samir said, I would challenge the slide you put up there, in the sense that the way that your trial design that you presented is advocating treating patients who are eligible for active surveillance, and I don't think that's our goal as treating surgeons, and that we shouldn't be trying to push that agenda in terms of delivering over-treatment.
Yeah, and I just want to be clear that we are not advocating that either.
Okay.
This is among the population of active surveillance patients that we've heard, some industry or investigators feel should be, offered additional treatment, and whether that population exists, I think is still an open question. We had an oncology advisory committee meeting for TUCAD, and certainly we heard multiple perspectives around how you define that population and the appropriateness for additional treatment, as well as some of the trial design considerations and limitations therein.
Thank you.
Rafael?
Rafael Sanchez Salas, I'm very glad that you mentioned the TUCAD, because the TUCAD trial is exactly what you presented there. They managed to do a comparison against active surveillance at the time with a population that obviously, as Dr. Watts alluded to, is not indicated for any kind of treatment. And then they evaluated the secondary treatment. There was a benefit on the focal therapy arm. So how do we account when we don't choose the right population, how the FDA accounts for technology evolution? Because now we know that the index lesion is there, it's not just hypothetical, that there is proven facts that is actually has an impact on natural disease. So it's just a trial, or it's just a device? Because when you look at the device, cryotherapy was approved. Fernando just mentioned that, and there's a line of other devices that need approval. So do we all need to go through the TUCAD trial in each one of the technologies?
Well, again, TUCAD was it considered a drug? It had an active therapy of a drug, so it fell under drug regulations, not device regulations. I think that if you watch that advisory committee, there were certainly a number of other issues that came up, around the design and conduct of that trial. I think the primary issue was the lack of follow-up of patient-reported outcomes after definitive therapy. So one of the open questions was, patients get a focal therapy and then get a definitive therapy. Is that what's going on longitudinally with patients after they receive that definitive therapy? Are you actually preventing morbidity? I think that ended up not being very well captured, and certainly was one issue. I think starting from pre-specified design and capture of the endpoints that I mentioned, I think probably may still require another discussion at an ODAC. I don't know, but certainly is potentially one path forward in that population.
I think I don't want to come off as saying that I'm opposed to the Weinstock design, because I think the intrinsic value of treating lower risk patients is yet to be understood, but it comes from the idea that still about 60% of those patients end up with active treatment, prostatectomy, or radiation at some point in their life, typically within 10 years, even in the PROTECT trial. And so the proposed benefit there is that you may avoid the morbidity of radical therapy in a population that has a very low risk of prostate cancer mortality over 15 years. So I think those trials answer an important question. But of course, the question we'd all also like to be answered is, can it be used as active treatment in a subset of patients who need active treatment, and can we better select those people? Brad, you have a question?
Yeah, thanks. Brad Rosenberg. Thanks for your talks. They were all very good. I have a question for Dr. Klotz. First of all, I learned something very interesting every time I hear you speak. It's great. And this is certainly one of them. I would love to believe that invisible MRI lesions are not significant, and I probably think that's true, but I think we've all had patients that have had MRIs where you have, let's say, a 8 to 10 millimeter lesion that's positive, and you biopsy, and that is positive, let's say, for grade 2, but then you also have 10 of 12 systematic cores as well that are positive. So my question is, first of all, do you think that those are biologically different, the invisible areas of grade 2 versus the non-invisible? And also, since this is a focal therapy meeting, would you even consider that as a focal therapy candidate, just the positive area and not the rest of the gland?
Yeah, so thank you for the flattering comment. It's early days, I would say, and any this is a general comment about the biology of invisible tumors. There's clearly outliers. So, but this is not it's not an all or none thing. So the patients with, who undoubtedly harbor significant cancers that you're missing because you're not doing systematic biopsies, I believe are going to show up over time. And as I mentioned in my talk, that's one of the things we have to prove. So I think, I don't do systematic biopsies. I do lots of focal therapy. I follow the patients. My belief, maybe it's an act of faith. I think it's not an act of faith. It's an inference based on interpretation of the data, is that those cases are going to show up at a time when the patients are still curable. So you haven't, and that's the small minority. Those really are the patients who have invisible, high-grade aggressive cancers are relatively few and far between. And of course, it's a trade-off. It's a classic, medical phenomenon. The benefit of not doing systematic biopsies, in the United States, probably 400,000 men per year could be spared biopsies if you don't biopsy the image-negative cases, is massive. And the potential downside, I think, is pretty small. So I don't mean to be dogmatic at all. Like, you have to be careful, and we need more data. But that's, at the moment, that's how it looks to me.
Brad, I think a lot of the patients, though, that you're talking about, the guy who has 10 cores of GG2, but a reported ROI in the left apex, if you really look at their MRI, they often have more diffuse change, but the radiologist has just chosen the more dominant part. So often, if you go back and look, there are abnormal imaging findings in the remainder of the gland. But I agree, those are unique and challenging cases. But as a follow-up to that, Lori, one of the things I love the story, invisible disease doesn't matter. But one of the things I really struggle with is, what does that exactly mean? Because on the one hand, you have the invisible disease that represents a focus of cancer, right? Contralateral, small tumor. On the other hand, there's a lot of literature, our own place and others have published, that MRI predicts sort of the core center of the tumor, but that there's a whole, as the UCLA guys call it, penumbra or halo of tumor that is largely MRI invisible. And we make a big deal about factoring that into our focal therapy treatment planning, because if you undertreat that tumor, that penumbra often becomes visible over time. So does that imply that invisible disease that's spatially related to visible disease is biologically relevant, but invisible disease that's not is irrelevant? And how do we figure that out? A lot of the studies you pointed at, it's interesting how they find invisible disease. It's sort of arbitrary. They'll pick a part of the gland that wasn't well seen versus a part that was, but the spatial relationship is not that well determined.
Yeah, it's a great question. Is the penumbra that looks normal in MRI biologically different from the part of the tumor that is showing up? And I don't know the answer to that. And I totally concur with the principle that because MRI tends to assess volume, that you need to treat significantly beyond the volume that you see to eradicate the cancer. So in a sense, I think like both are true. The issue is more to me multifocality than the actual volume of the index tumor.
Yeah, and I think in clinical practice, that's often hard to distinguish, right? So if we don't do systematic cores, then we make mathematical presumptions about the size of the index lesion, right? If we don't do, I think there's a talk coming up in the meeting on perilesional biopsies versus systematic, which I'm looking forward to. But I think, that's the question. If we just do 4 targeted cores from the middle, then we're guessing the extent of the tumor. But if we oversample, we're probably including multifocality. So it's a real clinical challenge.
And I think what you just mentioned, Samir, is critical. Because one thing is the posture that you have and the science behind it, but the other one is the practicality of what you want to treat and when you want to limit your treatment. For us, the negative biopsies, which come mostly out of systematic, gives us an extent to where we know there's no tumor and where we should set our boundaries on. So we can spare bundles better, prevent treatment extending into the bladder, neck, or the urethra, or anteriorly. And that is important as well. So it's not just the fact that you're focusing really on those areas that you know, at least based on several cores that are negative, so you can really set up your boundaries and go beyond the extent of what's seen on the MRI.
So can I just add, I think it partly depends, now we're getting into a different topic, which is your kind of philosophy of focal therapy.
Well,
how focal?
Exactly.
So I would say, yeah, if you are treating very focally, which I've moved away from that, I have to say, so, I usually, even the most focal cases would be approaching hemiablation. Yeah, I think there's something to be said that if you're just going to treat that index lesion and not the penumbra, you need to be bloody sure about what the margins of your tissue are.
The penumbra may be determined by the negative core, so we're never going to.
Yeah, but if you're planning to go beyond anyway, maybe it doesn't matter so much.
So I just have a follow-up question to that, and some of this comes into counseling your patients, which was a big topic of discussion in the morning session. So if you, in your practice, as you said, you only do targeted biopsies, a lot of folks in Europe are also doing the same. If you have a small focus, perhaps GG2, maybe GG3 contralaterally, okay, it's not seen yet on MRI, but theoretically that will progress over time. Would you rather know that up front in terms of being able to counsel your patient, or would you rather wait and have that declare itself over time? Some of this is subjective, but I'm just...
Well, I would rather, of course I'd rather know, but at what cost? And also, how many, you're talking about a small lesion, too small to show up on MRI, so you may not even hit that with your systematic biopsies. So, if you could get it as a freebie, yes, but it's not a freebie. It's going to involve biopsying so many men to find that one case, and you're going to find it eventually anyway. So if it was, it's the cost. It's a cost-benefit analysis.
All right, I just want to bring it back to the FDA, because Laurie and I could argue all day. I have so much to say on that topic, but that's not the point. We really want to get our partners from the FDA. So Dr. Anthonino, you sit here and listen to where we are in our specialty, and you've been here for, this is, I believe, your third year with us. And the transition of the conversation, the direction of the field, how do you view this with our goal of really proving or validating our approach and how we approach men with low to intermediate risk prostate cancer in the era of getting these devices to market? You're getting this, a 20,000-foot view, and then you get stuck listening to the minutia of us arguing on the genotypic, phenotypic expression of MRI visible lesion, and is this clinically significant or not?
Sure. Yeah, we're very open to have these conversations in detail. As we described earlier, there is a process, and we also have what we call the pre-submission process, which allows companies to come in, explain to us what their protocol might look like for us to get an early understanding of what the benefit and risk profile may look like. As Dr. Pinto alluded to, some of these are ethical questions, how we can treat these patients. We often rely on feasibility data to give us some look at what effective this might be before we begin an IDE, a clinical study, to understand more about what the benefit risk profile may end up looking like. It allows us to hedge in some regard. In some instances, we can put mitigations in place whenever we're unsure, such as stopping rules, to take a small number of patients and put very stringent rules in place to make sure that we're not getting into a situation where it's unethical and patients are being treated in some scenario like if we're not comparing to the standard of care, if they're more at risk. So there's a number of ways we can approach it to try to get research off the ground to support future marketing applications for high risk cancer treatment. But we're here to partner and to have these conversations in the least burdensome way as we can. And our door's always open to hear more about what protocols look like, what clinical trial design looks like.
So I have a follow-up for that. So that's completely the outline, the process. My question is can we break the problem into more subtle aspects? Like the first, what's an acceptable patient to enroll in a focal therapy trial? Because all the technologies are ablating and controlling disease. Can we agree on the subset of patients? So it quickens the process. And what are our secondary endpoints? Have we agreed on those? So we're just plugging in the approach in the middle, opposed to having to discuss who's a candidate and how does that candidate need to be treated and what are their secondary endpoints. Because for me, the ablation is it's somewhat similar between the technologies. We're trying to eradicate the tumor in the tissue in some manner. Is that a possibility to break these components out instead each company have to start over from the beginning to say what patient population, or there's a spectrum that we're just picking from column A, column B, and these are endpoints on column D?
Yeah, so one of our goals, it would be to create what we call a guidance document, which is our current way of thinking on how this would look for prostate cancer design in trials for focal therapy ablation. But that, we're not there yet. We have that for a tool-like claim. Some of the technologies are not as similar as I think you're alluding to them to be. So there's some differences that may raise different questions of safety and effectiveness. But it generally, we like to get there. We're trying to get there, but there's a lot of very specific detail in regard to the type of populations that are being treated, what their risk scores are, and we have to weigh that in the context of benefit risk. So again, we're open to having these conversations. The earlier, the better. We'd like to get to a guidance one day where we can have that laid out, but we're not there yet.
Thank you.
So on a different note, the de novo and 510K experience over 3,000 approvals per year overall. We saw that data, but for the PMAs, the pre-market approvals, we're talking about low numbers, 30, 40 trials per year. And in your experience on the device side, CDRH, how often do you see governmental support for long-term device trials for pre-market approval as opposed to industry-sponsored trials? Because I think one of the issues with the industry-sponsored trials, to achieve that long-term outcome may exceed the lifetime of the company. So what's your experience there? Do we have any government pathways by which we can seek long-term public funding of these device trials?
So there has, we're always looking for ways to augment our understanding of the risk-benefit of devices. There are registries. We look at real-world data consistently and try to figure out how we can use real-world data to augment our understanding. That's one way for us to gain more understanding and hopefully understand more follow-up and long-term data. So we're always looking to partner in that regard, and I think there are some efforts from a number of institutions that have started looking into registry-type data. And we've been trying to work in that regard for quite some time. We call it real world data.
Yeah, I think we're on we're kind of on time, so maybe it's a good time to wrap up. Thanks to all the speakers for a great session, great kickoff.
Really enjoyed your talk.
Thank you.
I need to do something about the